For meiosis, we're starting with the same model organism that has two different kinds of chromosomes. The long chromosomes, yellow and red, are homologous to each other. The short chromosomes, blue and green, are homologous to each other. In this cell, it is diploid because it has two of each chromosome, two long and two short, and it has 4 total chromosomes, 2 long ones and 2 short ones. During G1 of interphase, this cell has 2N DNA. It has 2 copies of every gene, one that came from the mother and one that came from the father, one on each homologous chromosome. Prior to cell division during meiosis, the cell has to go through DNA replication. So just like during mitosis, to prepare for meiosis, the cell goes through S phase of the G cycle and of the cell cycle and replicates its DNA. Now each chromosome is made of 2 identical copies called sister chromatids. Again, just like in mitosis, prior to meiosis, the centrosomes have to replicate. Now the cell is ready to begin meiosis 1. The DNA condenses, the nuclear envelope begins to break down, and the centrosomes begin migrating as the microtubules grow to form the mitotic spindle. As meiosis continues, we're still in prophase of meiosis 1. The homologous chromosomes find each other. They line up and are joined together in a process called synapsis. Towards the end of prophase, non-sister chromatids from homologous chromosomes cross over. Now the sister chromatids are not identical to each other. These chromosomes are now recombinant. They have a combination of DNA from this organism's parents. In metaphase of meiosis 1, the chromosomes line up along the metaphase plate. In anaphase of meiosis 1, it is the homologous chromosomes that separate from one another, while the sister chromatids remain joined at the centromere. At telophase of meiosis 1, each cell that is produced is now haploid. There is only one long and one short chromosome in each cell that is produced. But each chromosome is made of 2 chromatids, and so the amount of DNA in each cell produced is 2N. Meiosis 2 begins without DNA replication happening. We've gone through prophase of meiosis 2 and are now at metaphase of meiosis 2, where the chromosomes are lined up along the metaphase plate. As the microtubules shorten, the sister chromatids are separated from one another, similar to mitosis. The sister chromatids that are separated from one another, however, are not identical due to crossing over that occurred during prophase of meiosis 1. During telophase 2, the DNA decondenses, the nuclear envelope reforms, and the cells pinch apart by cytokinesis. The cells that are produced are both haploid and 1N, with 1 copy of each gene and 1 of each chromosome. Each cell that's produced has a unique genotype due to crossing over. You can see the cell in the upper right-hand corner is made of an all yellow and all green chromosome. The one below it, however, has some blue genes on the green chromosome. On the cell in the upper left-hand corner, one chromosome is all red, where the blue chromosome has some green genes, and the recombinant chromosomes in the cell on the lower left-hand corner are cut off.